So we were discussing about how resting membrane potential is developed inside a myocardial cell and how does it sets the stage for the development of action potential. And then we discussed how action potential is being generated inside a cell. What are the different phases involved in the graphical representation of the action potential? What are the ion channels involved and at what stages, at what phases, what channels are involved and how does it reflect on the action potential curve? So now we know how resting membrane potential is developed and how that sets the stage for action potential and how action potential is being generated in the myocardial cell. And we have also learned how is the action potential, the electrical action potential is coupled with the mechanical action of the cell, which is contraction of the sarcomere unit, resulting in muscular contraction or systole. Now, we need to learn how this action potential is being transmitted or is being communicated from one segment of the cell to the other segment of the cell, or sometimes even from one cell to the adjacent cell. So we know, just to recap again, the resting membrane potential generated in the cell because of all these processes were minus 90 millivolt. Because of the cation influx or stimulation of the cell, it was raised to minus 70 millivolt, which has resulted in the opening of the voltage gated sodium channels. And because of the influx of sodium, the potential, electrical potential inside the cell was increased towards positivity, which means the cell was in the polarized state, it became depolarized in terms of electrical voltage inside the cell. And the opening of the sodium channel was for a very brief duration, and immediately after this brief duration the sodium channels are now closed, which means there is no more cation loading onto the cell, and at the same time we have potassium channels being opened. The voltage gated potassium channels are opened. So because of the closure of the sodium channels, there is no more ions coming in, positive ions coming in, and because of the opening of the voltage gated potassium channels, the cell is losing cations. We see a dip in the action potential. It is coming towards negative, but as soon as the calcium channels are being opened, it is balanced. The ion exchange is being balanced. You have potassium going out, but simultaneously calcium coming in, which means there is no net change in the electric voltage. It maintains a plateau phase, and by the end of the plateau phase, we see that the calcium channels closes, but the potassium channel remains open, which means the cell ultimately loses cations. It becomes, the potential inside the cell dips down and it touches the resting membrane potential. And because of all the actions of the sodium calcium exchangers, the action of the sodium potassium ATPases, the ion exchanges taken place during the various phases of action potential are being reversed, and the cell re-establishes the minus 90 millivolt resting membrane potential during its resting phase. So it re-establishes the resting phase, it re-establishes the polarization, which is called as repolarization by the end of action potential. So this is what we have learned. Now we must learn how this action potential generated in this segment of the cell will be communicated to the adjacent segment and then to the adjacent cell. Now here we know in the phase 0, what is responsible for phase 0? It is the opening of the sodium channels that are responsible for the phase 0. So when the sodium channels open, there is some amount of sodium is entering from the extracellular compartment to the intracellular compartment. So we know this is the cytoplasm, the cytosol, So in the intracellular compartment, few sodium ions that are influxed through these voltage gated channels will migrate to the adjacent segment of the cell. Few sodium ions will migrate to the adjacent section of the cell, and entry of this sodium ions will slowly increase the resting membrane potential of this part of the cell, which is minus 90 millivolt. The resting membrane potential at this part of the cell is minus 90 millivolt. Because of the entry of this sodium, the resting membrane potential here will be rising to minus 70 millivolts. So here is this minus 90 millivolts. So because of this entry, here we have minus 70 millivolts. So as soon as the electric potential reaches minus 70 millivolts, what happens? The entire mechanism will be replicated here, because the voltage of minus 70 millivolts will result in opening of the voltage gated sodium channels. So sodium will be coming in for a brief duration, and then we will have potassium going out, and then the potassium will continuously go out, and then at this phase, in the plateau phase, we will have calcium coming in the plateau phase, and by the end of the plateau phase, the calcium incoming is stopped, and the entire thing repeats. So what happens? We have another wave of action potential being generated. As sodium comes in, it goes to depolarization 0, and then you have stoppage of the sodium channels and opening of the potassium channels. So you see the dip, and then you have opening of the calcium channels along with opening of the potassium channels. It maintains the plateau phase, and as the calcium channel closes, the potassium channel continues to leak. So the cell repolarizes back to the minus 90 millivolt. So this segment, depolarization followed by repolarization of one segment happens, and during the sodium that enters during the depolarization of this segment migrates to the adjacent segment and depolarizes the adjacent segment of the cell. This is how this action potential is communicated. So what happens when this phase, this particular segment of the cell, I shall separate it like this. What happens when this particular segment of the cell is depolarized? When this particular segment of the cell is depolarized, this particular segment is undergoing repolarization. So what we have here effectively is a wave of depolarization and repolarization. Again, depolarization and repolarization. So a wave of depolarization and repolarization sweeping the contractile myocardial or sweeping the myocardial membrane is what we call as action potential. Is that clear? So just to repeat once again, in the first phase we have when the cell is being stimulated, the action potential, the resting membrane potential is raised to minus 70 millivolts, which results in opening of the voltage gated sodium channels, so that it moves to phase 0. The cell depolarizes. Some amount of sodium from this phase, from this area, moves or migrates to the adjacent area and increases the resting membrane potential over here. It increases the voltage from minus 90 millivolt to the threshold potential, which is minus 70 millivolt, which will result in the opening of the voltage gated sodium channels of that particular segment and initiates an entire cycle of action potential at that particular segment. So this is how it is being continued. Now what happens further? We have another cell adjacent to this cell. We have another myocardial cell here, and we all know that these myocardial cells are connected by special structures between them. We call them gap junctions. we have fluid filled gap junctions in between these structures. So these gap junctions, we understand, they permit free flow of cations. So what happens? So when an action potential, when the sodium gate is opened here, when the threshold, when the membrane potential touches the threshold potential at this particular segment, the voltage gated sodium channels are opening at this particular point. It depolarizes this part of the cell, and simultaneously some amount of sodium migrates to the adjacent cell, which will result in increasing the resting membrane potential of this cell from minus 90 millivolt to the threshold potential, which is minus 70 millivolt, which will ultimately result in opening of the voltage gated sodium channels in the adjacent cell, and it will trigger an entire cycle, initiation of an entire cycle of action potential over here. So here we have the action potential being generated in the adjacent cell, which is phase 0, phase 1, phase 2, phase 3 and phase 4. So it goes. So this is how the action potential is being transmitted from one segment of the cell to the adjacent segment, or from one cell to the adjacent cell through the gap junctions. So this is how it is conducted, and the process of, or the wave of depolarization and repolarization sweeping the membrane of the myocardial cells is what we call as action potential. So now we know how is action potential generated, how is resting membrane potential generated, how it sets the stage for action potential, and how an action potential is generated, and how this action potential is transmitted to the adjacent structures in the myocardial cell. In the beginning of this lecture, we have mentioned different types of tissues in the heart. We said we had specialized cells and contractile myocardium. What we have discussed today is the generation and conduction of action potential in a contractile myocardium. Now we need to discuss how does the generation and conduction of action potential happens in a specialized cell. Typically, for example, SA nodal cell. They are not same, they are different. So let us see what difference they make.